We're pretty excited about it because it was really a phenomenon-based approach that developed out of an observation. Innovative Cellular Therapeutics had originally developed a CD19 CAR product. It was an armored CAR, so it had a PD-1 dominant negative gene included in it. And it did quite well in clinic and easily would have competed with Kymriah or Yescarta. But by the time we were ready to move to the U.S. with it, it would have been an also-ran essentially. And we were seeing some initial activity in solid tumors that we wanted to follow up on. And the difficulty, though, was that we weren't seeing a lot of activity. So we wanted to make it better. And in looking at our CD19 product and some of the clinical profile of the activity there, we realized that the CD19 CAR, actually the activity of that product caused the rest of the T cells to also proliferate systemically, which now I don't think is a novel observation. At the time, there wasn't much indicating that that would be the case or is the case. We realized that we could potentially utilize that effect in order to try to promote proliferation of the solid tumor CARs. And the reason for that is because the main barrier for solid tumor CAR, or really any T-cell immunotherapy for solid tumors, is the ability to get the T-cells into the tumor, because the tumors and the target are localized in that tumor tissue, so they're not available systemically. The CARs have to make their way there, engage, and then start their activity, which can be difficult in the tumor environment. So what we realized was if we utilized the CD19 CAR, that we could get that activation sequence going systemically and prior to the infiltration of the solid tumor by the solid tumor targeting CARs. So we call it coupled CAR because we really coupled the activity of CD19 to, or the CD19 CAR, sorry, to the solid tumor CAR as well. So there are, eventually we developed sort of refined versions of that had higher activity, and now we're seeing clinical efficacy. So it wasn't the magic bullet entirely to do that, but that was certainly the basis for developing the current version, which we think has pretty good clinical efficacy in our, at least in our lead target, which is colorectal cancer.